A little background information about what stem cells actually are. Stem cells are non-differentiated cells that divide through mitosis. There are two different types of stem cells: adult stem cells, which are found in various tissues of humans, such as the bone marrow, and embryonic stem cells, which are derived from epiblast tissue of the inner cell mass of the blastocyst, which is an early stage of a developing embryo. Embryonic stem cells can be transformed into specialized cells through cell culture. Groups of embryonic stem cells are known as cell lines because the genetic material comes from the same fertilized egg. This is what the embryo looks like when the inner mass cells are taken to form a new cell line. Only embryos that are 5 to 6 days old are viable for the creation of new embryonic stem cells. Embryonic stem cell research should be allowed to develop without limitation or restriction set by the government. Federal funding should be utilized to ensure that America is at the forefront of stem cell technology. Unlike embryonic stem cells, adult stem cells are derived from tissues of mature adults. Adult stem cells can only divide into the type of cell that they originated from, unless they are induced pluripotent stem cells. In that case, the adult stem cells can be induced to produce cell types other than the one from which it was derived. However, iPS cells have certain drawbacks. Scientists that have worked with these specific cells believe that, if injected into human patients, they may divide uncontrollably and cause cancer. Embryonic stem cells are pluripotent, meaning that they can develop into every cell type, including the endoderm, which forms the intestines and lungs, the ectoderm, which forms the nervous system and skin, and the mesoderm, which forms the muscles, bones, and blood. In order to be considered embryonic stem cells, cells must exhibit two properties: self-renewal, or the ability to complete multiple cycles of cell division while undifferentiated, and potency, or the ability to differentiate into specific specialized cell types. Adult stem cells exhibit unipotent characteristics, meaning that cultured cells can produce only the type of cell from which it was derived. Much more versatile than adult stem cells, embryonic stem cells exhibit pluripotent and multipotent characteristics. Pluripotent cells can differentiate into almost all cell types, while multipotent cells can differentiate only into a closely related family of cells. In order for stem cells to be utilized for their full potential, scientists must give the embryonic stem cells and stem cell technology legitimate chances to advance. Embryonic stem cell experiments are extremely new. Specifically, embryonic stem cells were used for the first time in human patients in October 2010. Sponsored by the Geron Corporation, the results of the tests look promising, but scientists have no way of knowing the ultimate effects of the experiment. If embryonic stem cells were limited by restrictions on federal funding, the scientific community could not witness the potential benefits of utilizing stem cells to treat deteriorating diseases, such as leukemia, Parkinson's disease, multiple sclerosis, and numerous spinal cord injuries. Even if scientists discover that embryonic stem cell experiments do not effectively cure the aforementioned diseases, information gained from studying the reactions of the immune system and other functions in death will enlighten scientists in other ways. During the Bush administration, President George W. Bush approved federal funding on existing embryonic stem cell lines that no longer had the possibility of developing into human beings. The National Institutes of Health composed a list of the viable embryonic stem cell lines, known as the Human Embryonic Stem Cell Registry. Reversing the Bush policy in October 2009, President Barack Obama issued an executive order entitled Removing Barriers to Responsible Scientific Research Involving Human Stem Cells. The executive order altered the way the National Institutes of Health can conduct stem cell research. Obama allowed the use of federal dollars to fund expanded human embryonic stem cell research. But that's not what I'm here to talk about. I'm here to talk about having an amazing class in biology, and I'm going to miss it. I'm going to miss you guys. I'm going to miss Mrs. Frost. I think I'm going to sleep.